Oh, for this particular
this project. So he's put there as one of our artists in residence, right?
Is it clear or is it?
No.
Oh.
Just,
I mean the project.
This is actually a site. That's why I was an architect, and I was going through some the project we started
Oh, so you have this screen?
Yeah, I just had it print all over. Yeah, it's
excited, but I reminded her that
sweatshirts Here we go. No, I mean, 5 or 10 minutes or something. Yeah, John. Yeah, everyone gets some talk out of it. Yeah, just in the nick of time.
We're gonna start in 5 minutes at 7:20. So make sure you have a full beverage. There's beer, wine, juice, and waters in the back, and snacks, and then have a seat. We're gonna start at 7:20.
The teleprompter. It's going.
A video camera?
Okay, so I'm going to talk to you about that later. I was
Huh?
going to bring it up
Sorry.
while the installation was still happening. Cool. Is it Bluetooth or is it?
Yeah, it's Bluetooth, and you can control it with your phone. It can record onto SD card, micro SD card, and then also
Okay, cool.
stream live.
This proposed change was a collaboration with
We just got it for the department.
So it came in
Yeah, I think we should do
yeah,
that.
so there's a
I'll show you.
so part of what I do is pictures of the canal that have. Oh, that's perfect. Amazing.
And then you can. That's what the camera sees, and then this is the whole field of view.
Oh, okay.
And then you
Can
cut
we,
the camera.
can we do it to include
Yeah,
the
yeah.
screen?
Yeah.
Yeah, perfect. Great. That's awesome. Oh my God. Neat. Approach the idea in their products visually and to use more of the material. No, I mean, in case people or in case it was that study
I try to use it, but
It's like this and
what?
I know.
Hello, welcome to the BFL. My name is John Haida. I'm the director of the lab. Thanks for coming tonight. I'm really excited to be presenting for you our latest artist in residence. It was a special artist in residence who actually worked from last spring through the summer and is now presenting for you a project that they've been working on for some time. I would assume that you'll be talking a little bit about that through this process. So without further ado, I want to introduce Elizabeth Hénaff. And are you guys talking about today? But you're definitely part of the project. So yeah. So in order of height, so this is Dr. Elizabeth Hénaff and Quate and Matthew Seibert, and they will be presenting their work as BKBioreactor for you today. Thank you. Thanks.
Cool. Thanks, John. Can you guys hear me okay?
Yeah.
Okay, awesome. So yeah, I'm Elizabeth. I'm a computational biologist at the Weill Cornell Medical College. And I don't think I've had as much fun as I have over the last couple of months spending the summer here. So this has been really an amazing experience to be artist in residence here at the VFL and actually make stuff with my hands and not just program a computer all the time. So I'm super excited to tell you guys tonight about the kind of background and motivations of this project and then the stuff that we've been making here at the VFL. So I'd like you guys to take a moment and conjure up a mental image of what you think when you think of the word environment.
Think of.
Environment, the word environment. So, I mean, when I think of environment, I think of maybe, some kind of bucolic landscape with like trees and plants and animals and stuff. But for better or worse, the impact of humans on the environment has changed it quite drastically, and we've contaminated a good bit of our environments. And so, the EPA has a name for that. Oh, do I have a pointer?
Pointer?
I can point.
Yeah. Let me check.
Yeah, no worries.
Don't wait for me, but if I
Okay,
find it.
cool. I'll walk around. So for better or worse, a lot of the environments that humankind has interacted with have become very contaminated. And the EPA, which is the Environmental Protection Agency, has this special program called the Superfund program, where they designate certain sites as priority for remediation. And that priority is mostly based on the desire to protect human health and the environment by cleaning up polluted sites. And you would think that that might be something pretty rare, but actually if you look at a map of the United States with the Superfund sites that have been annotated, they're all over the place. And somewhat correlated to the first industrialized states, maybe less so in the middle, but they're quite ubiquitous. And if you're curious, you can look where you live, and you can see that we've got two of them here in New York City, specifically in Brooklyn. So this is the Gowanus Canal, and this is Newtown Creek. And so it kind of hits home when you have a Superfund site that's like basically in your backyard. This is the Gowanus. I live like here, and so the Gowanus Canal was not always a Superfund site. It used to be a creek, actually. And so what we're looking at here in kind of lighter blue is the outline of the original Gowanus Creek. And then it was dredged into its present-day shape, which is all kind of like angular and straight, in the 1850s. And so the original goal was to dredge a canal to serve as a transportation system to and from the factories that were in operation around there. But not only did it serve as a transportation system, but it also served as a de facto dumping site. And so these different factories would just dump all of their waste into the canal, it disappears underwater, doesn't exist anymore. So that's what led us to the Superfund site that we have today. And so there's various different industries that were in operation around there. There was a coal tar extraction factory, which is actually the source of the main part of the contamination. So there's a maybe 3 to 5 meters, so 10 to 12 feet of like viscous tar-like sediment that has accumulated at the bottom of the canal. There was also paint making factories, tanneries, soap making. All of these have contributed to this particular cocktail of toxicity that is the Gowanus Canal at the moment. And so this is maybe the most unenvironmental of environments that you can think of. You wouldn't even think twice about wanting to do something to this environment. But the question that arose that actually has motivated this project, which is the question posed by Ian, was what's going to happen once this Superfund site is remediated. So the kind of standard modus operandi for the EPA for this particular type of contamination is a mechanical remediation. So they'll empty out all the water, dredge the contaminated sediment that they can, take it out, treat it elsewhere, cover the canal with a concrete cap, put the water back in, and then say that everything's okay. So then you can take your water quality measurements, and Whole Foods that's built right there can have no problem stating that they're the greenest Whole Food in the state. But what's going to happen is that mechanical disruption is going to destroy the environment as it is. And so, our fearless citizen scientist, Ian, posed the question of, well, what is this environment at the moment now, before it gets destroyed? What could possibly be living in there? We don't see many plants or animals living in the sludge, so it's probably bacteria. And if there is something living in there, then what's it doing? So we set out in these like pretty cool expeditions. Actually, it's a lot of fun. We get to wear hazmat suits, which always makes you feel like a badass. And so we've constructed these kind of DIY sampling devices, which consist of lengths of PVC tubes that you can see here that are fitted with kind of like a flexible ends. And then we paddle around in the murk and then take samples by driving the PVC tube into the sediment and then capping it. And with the same principle as sampling a drink with a straw, where you put the straw in the drink and then you cap it and then you can pull some liquid out, we pull out some sediment. And the sediment is this like black tar viscous stuff. We have some on display next door, so you'll be able to see it. So then we take it back to the lab. In this particular case, we were also working with JetSpace, which is this community biohacker lab in Brooklyn. That if you guys aren't familiar with it, I would highly recommend checking it out. It's a full-blown molecular biology lab. It's not affiliated with any academic institution. So as long as you get training, you can become a member and conduct your own scientific experiments. It's awesome. And so there we would do the DNA extractions and then take it back to my lab at Cornell, where we would sequence the DNA and do the analysis. And so the experimental design was as such. We selected 14 different sites along the canal to sample. So these sites correspond to different conditions of light and darkness. Some of them are underneath the bridge, some of them are exposed to light. Different conditions of salinity also. This is the tidal system, so you have tide coming in from the East River twice a day, and there's still like fresh water input from the watershed, so you have kind of a gradient of salinity, different conditions of turbidity also. And so we take these samples. We've been doing that seasonally for the last couple of years, and asking the question of what bacteria are in the sludge, what are they doing, And the hypothesis we're trying to test is whether they might hold any key to bioremediation, because everything that can live in such a toxic environment has had to develop some kind of mechanism to respond to that challenge. So I'm gonna go over a brief overview of like the methods that we use for that. So we start with our test tube with a mixed bag of bacteria. So we have different kinds of bacteria, different species, which are represented here by the different colors. And those species are present in different relative abundances, which are represented by, there's 2 red ones and 4 purple ones. And so what we do is we extract the DNA and sequence it. And so we're going from a mixed bag of bacteria to a mixed bag of pieces of DNA that belong to these different bacteria. The sequencing technology that we use is called Illumina sequencing. What's particular about it is that you can only read about 100 or 150 layers at a time, which is a very small piece of a bacterial genome that's anywhere from a few 100,000 to a few million letters at a time. And so then the job that we need to do computationally is try to reconstruct and find which species these little pieces of DNA belong to. So we do that by comparing them to reference genomes. So there's some bacteria for which we have the full genomic sequence. And so if we find a piece that maps unambiguously to one of these genomes, so it maps to one and only one, we're going to say that that genome is present. If we have lots of pieces that map unambiguously to a genome, we're going to say there's many individuals of that particular species that are present versus a few individuals of another species. So that allows us to identify which species are there, so that's species classification, and what their relative abundance is. So here we have multiple dimensions of data. We have species description, so classification, quantitative relative abundance, and also an intrinsic geospatial dimension because these samples have been collected at different specific locations. And there's many different ways how you could represent these multiple dimensions of data. One of them is like this. So here what we're looking at is a phylogenetic tree that describes the relationship, genetic relationships between all of the different species that were identified over the entire set of samples. So these 2 guys that are close to each other in the tree are more related to each other than these 2 guys who are very far from each other in the tree. Then what we've represented here is the distribution and abundance of these bacteria. So we've color-coded in these concentric rings whether the bacteria is present or not in each location. So we're starting here with the upstream location, and each concentric ring represents a different location all the way to the most downstream location. So these guys, for example, and the abundance is color-coded by color intensity. So these guys, for example, were present in a very high abundance everywhere, and this guy was present in low abundance in just one spot. And so this is informative as long as you know something about these bacteria. So some bacteria have been very well studied, we know a lot of things about them. Some bacteria have not been very well studied, and we know barely anything about them. But we can pick out a few that are interesting. So this guy, for example, is a sulfate-reducing bacteria. We know it decomposes carbon-based materials. That's interesting with respect to our hypothesis. It lives in an anaerobic environment, which coincides with the marine environment. There's low concentrations of oxygen. It's also been classified as a marine organism, so that makes sense with the system being a tidal system. This guy is a methanogen. It's most often found in the human gut. So that makes sense because there's actually a good amount of raw sewage overflow that ends up in the canal when there's heavy rains. So the sewage system gets overwhelmed, and we have raw sewage that gets dumped into the canal. This guy is called Pseudomonas, and it's very well known actually for digesting organic pollutants and solvents. It's used sometimes in oil spills, for example. And so this tells us what bacteria are there, and to a certain extent it tells us what they're doing, as long as we know what these bacteria do. But we don't know that for all of them, and also bacteria are able to exchange genetic information very readily, And so a bacteria that has been attributed a particular function might have lost it since, or might have gained new functions that weren't identified for it yet. So in order to answer the question of what the bacteria are doing, we performed a different kind of analysis, which is called a functional analysis. So taking our little bits of DNA information, instead of comparing it to unique regions of genomes, we compare it to gene sequences. And so I don't know how far back Yale's last biology class was, but complex biological functions require the concerted action of multiple genes. So the alcohol in this beer that I'm drinking, in a few hours is going to be broken down into water and carbon dioxide, but that's not going to happen in just one step. It's going to happen through multiple different intermediates. And each intermediate reaction is catalyzed by a different enzyme. So there's many different steps, many different enzymes. Each enzyme corresponds to a different gene. And so that set of genes together that corresponds to multiple steps of a reaction is called a pathway. So what we did was a pathway analysis. We were looking for what complete pathways, what genetic functions were encoded in this data. And first, looking for kind of simple things, we wanted to know how these bacteria respirate, how they breathe. So also going back to your biology classes, there's two different kinds of respiration that we can do, is anaerobic and aerobic, meaning in the aerobic in the presence of oxygen and anaerobic in absence of oxygen. We're able to do both. So we prefer aerobic respiration. That's why we breathe. But when we exercise too much and our muscles get sore afterwards, that's because we had to transfer to anaerobic respiration because our circulatory system wasn't able to deliver enough oxygen to our muscles. So we're able to switch between the two. And so what we can see here is the abundance of these different pathways. So aerobic respiration, anaerobic respiration, aerobic respiration, in the 14 different locations here that correspond to the different spots on the map. And their intensity is color-coded from light blue, which is low, and dark purple, which is high. So we can see here generally that we have a lot more anaerobic respiration than aerobic respiration. This makes sense because this is an aquatic environment, and there's a lot less oxygen present in an aquatic environment than in the air. And so then we started pulling out to see if we could find pathways that would be interesting with respect to bioremediation. So do these bacteria contain pathways that enable them to degrade some of these toxic compounds? And we actually found a bunch of them, and it was super exciting. And so these are just a few of them. But what was exciting is that we were able to relate all these different pathways to historical data of different factories that have been in operation around the canal since the 1850s. So pre-sol degradation pathway, this is degradation of complex hydrocarbons. So this corresponds to the kind of contamination that would have been generated by the coal tar factory. Arsenic can be a byproduct of the soap making and tannery industries. Toluene is an industrial solvent and would also correspond to paint making industries. Atrazine is a component of herbicides, and we think is probably coming from runoff from the neighborhood. So we were super excited when we found this. This is amazing. This is the silver lining of a very dark cloud. And I think it's particularly elegant that by studying the genetic content of the microbiome, or the population of bacteria that lives in this environment, we have this kind of molecular trace of the history that this environment has gone through. So looking at the present day genes that are encoded by these bacteria, we're able to see what has happened to this environment. So I find it particularly poetic and beautiful. The thing is that obviously these bacteria aren't doing it fast enough. The canal is still contaminated. And so what we've been doing now is thinking about ways that we can accelerate that process. Yes, we have a website. So you can browse all of this data online. So part of this being a citizen science project is we felt it was really important to make the data public. So we have a website called BKBioreactor.com. You guys should check it out. And so it's interactive. You can click through the different species and see where they're distributed. And so the conclusion so far is that we know the microbiome of the canal looks like both the marine environment and the human gut, and that the population encodes some really nice bioremediation functions. But it's not doing it fast enough. And so there's kind of two different ways that we can approach this question. So we can either try to modify the bacteria themselves, so genetically engineer them to produce some of those pathways more quickly, or produce more of some of those pathways to be able to metabolize more quickly. That's something that we could do in an isolated environment. So say some of the sediment was dredged, and then you could treat it in isolation, like in a bioreactor or something. Or we can think of ways of modifying the environment of the bacteria themselves, so more of an in situ solution, because we can't release genetically modified organisms into the environment for perfectly good reasons. And so we can think of ways, instead of modifying the bacteria, to modify their environment. And so I'll go through kind of the sets of experiments that we've been doing for these two approaches to the same question. So one of the issues with modifying the bacteria is that the two computational analyses that we did, the species classification and the functional analysis, use kind of different parts of the data, and so it's hard to attribute a particular function to a particular species, because the bits of information that we have are really short, so they only provide us one kind of information, but not both of them. But there's a new kind of technology that has come out called the MinION. This is a DNA sequencer, and it's smaller than your iPhone. It's pretty awesome, and I think this is a movie. Can we play it? It has cool blinking lights. The future is now. You're going to be able to take this anywhere with you, like on a hike to whatever remote region in Antarctica, on a sailboat in space. We sent one of these guys up to the ISS, and astronauts have sequenced DNA in space. Like, this is super awesome. And what's really awesome about it is that it gives you really long reads of DNA. And so with those long reads of DNA, you can recover full gene sequences, and with that, you can attribute them to specific species. So we've done that for some of our data. I'll pass you the biological jargon of what these genes mean, but this is one of the genes that is part of the cresol degradation pathway. So we were able to recover the full gene sequence, and this is the kind of information that you need to then start tweaking those gene sequences to optimize them. And we are also able to attribute that pathway to a particular species. And so that's super interesting too, because like that, we can pull together information of what all the other genes that species has. And so that's one approach to finding out who's doing what, is getting enough information to do so. Another approach is doing culture experiments. So what we're looking at here are petri dishes. This is what we grow bacteria in. And so a petri dish is a base of a nutritional medium. So it's got all sorts of like amino acids and vitamins and stuff the bacteria need to grow. And you mix it with this like gelatin substance, so it's kind of squishy. And you pour it in this plate, and then you streak your bacteria on top. And like that, the bacteria have a substrate to grow on. And so what I did is I made different plates with, this is standard kind of media, so just regular stuff. This is media that I complemented with 2% of the toxic sludge from the bottom of the canal. This is media that I complemented with 20% of toxic sludge. So this experiment tells us a couple of things. Then I took samples from the canal and streaked them on it to see what kind of bacteria would grow. And so it's kind of hard to see in these images, but there's actually, not only are there bacteria that are growing on all three of these plates, but there's actually a lot more bacteria that are growing on the plate with more sludge. So that's super interesting because that means that some of the bacteria need these compounds to be able to survive. So that means that not only have they adapted to just be able to survive in this toxic environment, so with these very complex hydrocarbons, but they've probably evolved pathways that enable them to break them down, and that's what they use as their carbon source. So not only are they just surviving it, but they're actively breaking it down and they require it. So now we can take samples from these guys that are growing on the more toxic plate and then separate them out and then figure out what individual species they are. So we have some of these plates in the gallery exhibit with a microscope. You guys can look at it. They're in these pouches. Do not open the pouches. Because for two reasons. So these pouches there's like this little bag in it. It creates an anaerobic environment. So it's like a little chemical compounds that will absorb oxygen and release carbon dioxide because that's what these guys want to live in. But also they're hermetically sealed because we don't want for you guys to be breathing any mutant canal bacteria. So feel free to look at them, just don't open the pouches. So that's the experiments that we've been doing in the direction of modifying the bacteria. And now I'd like to talk about what we've been doing about modifying their environment. And that's specifically what we've been working on here at the VFL. So let's talk about the built environment for just a second. So we like to build buildings and structures to exist in, to live in, to sleep in, to work in. And we've got basically two choices as far as design of the built environment. We can choose the materials that we use, and we can choose the shapes that we build them into. And most of the time what we want to do is optimize human comfort. So the optimal temperature for humans is 19 degrees Celsius. So we'll want to be in a comfortable temperature range, for example. So optimal temperature for humans is 19 degrees Celsius. And then that will inform certain choices with respect to the environment. So we'll choose adobe houses with very small windows to optimize temperature in very hot environments like the desert of the western United States. Another metric that we might want to optimize against is acoustic comfort. So there's a certain level of decibels that are too loud, that's uncomfortable. And so we'll choose a particular sound refractive material to line our highways with so that our residences are not contaminated with too much sound. Or we'll choose to design a concert hall with very specific shapes so that we have an optimal acoustic experience. And so for this particular project, we're not wanting to optimize for human comfort, but we're wanting to optimize for the microbial residence of the canal. And specifically, we want to optimize for bioremediation activity. But the choices we do have are still the same choices, meaning choices of materials and choices of shapes. And so we looked to nature to see if we could find some examples where the choice of materials or shapes will influence the type of bacteria that live in an environment. And it turns out that both sea sponges and sea corals live in symbiosis with a whole host of bacteria, and we'll have different types of bacteria that inhabit different shapes, different types of cavities within the sponge. So there's some bacteria that like to live in these little channels here. There's other types of bacteria that like to live in the big cavities. And so that's an example of how the three-dimensional structure of an otherwise abiotic or non-living environment can select for a particular type of bacteria. It's been hypothesized that particular selection is due to water flow rate. So the small channels have a higher water flow rate than the large channels, and that some bacteria like more or less water flow. As far as materials choices, most materials that we use in the built environment are chosen for their antimicrobial properties. So cement has a very high pH that inhibits microbial growth. We treat wood with creosote so it doesn't rot. We put triclosan in drywall so that it doesn't rot. But I'm starting to understand that eliminating all the bacteria in our environment can be a bad thing. That's actually been associated to all sorts of autoimmune diseases, asthma, things like that. And so there's been more and more research that has been done towards developing bioreceptive materials. So materials that can potentially host microbial life. And there's a scientist, Sandra Manso, who's now at the University College of London Bartlett School of Architecture, who has been doing some really interesting research with different formulations of concrete. So concrete has a very high pH, and so she's been trying to bring that pH down without impacting the structural integrity of the material, so it can still be used as a building material. And so she's been able to develop these bioreceptive concretes that are more amenable to, or that don't inhibit microbial life. So with this in mind, we've been doing a bunch of experiments here, using like digital fabrication techniques and stuff like that to experiment with combinations of materials and shapes to see if we can find a combination that will optimize for bioremediation. And what we're trying to do here is directed evolution. So that consists in altering the environment of an organism to select for a particular function. And so the setup that we envision is this kind of setup. This is a bioreactor, like standard bioreactor in a lab. We test a combination of materials and shapes, assay things like toxin degradation rate, biofilm formation, and it came as really a revelation to me when I took a synthetic biology course a couple of years ago called How to Grow Almost Anything. So it's like synthetic biology and like biofabrication course, and so it came to a revelation to me that I can make my own experimental instruments, and so that's how I ended up here. And so I've been learning a bunch of digital fabrication techniques, like 3D printing. I guess you're cutting.
No, I know. Exactly. So cool. Actually, just as easy as like printing a file. It is.
So yeah, that's the wonder that comes with it. And then, so we've been doing a bunch of studies with different types of materials. So here, experiments with terracotta, with cement. I've also been extracting some silk fibroin in the lab, so you can mold nanoscale structures with silk proteins. That's pretty cool. And so the idea is to kind of mimic the sponge idea, so provide a network of different types of cavities with a quantifiable complexity, and do some experiments and see which materials and which shapes work better. And so kind of starting with this idea of how you have different sponges of different complexity, but not wanting to fabricate many different implementations of different complexity, we came up with the help of Tak and other people here with this tessellated design. So there's 4 different elemental blocks with increasing levels of complexity. So here we have 1 hole, 2 holes, 3 holes, 4 holes that then you can extrude and fabricate in 3 dimensions. And then you can assemble these using different combinations of different types of blocks. So you can assemble these into structures of arbitrary complexity and quantifiable complexity. And you can assemble these in 2 and also 3 dimensions. So you can go from a small experimental scale to a large architectural scale using exactly the same design. And so that's kind of the design process. We want to put them in a bioreactor. This thing costs like 20 or 30 grand. And so if you want to run many parallel experiments, I can't convince my boss to buy like 50 of these. But in the spirit of making your own instruments, I made my own bioreactor. And so these are a set of sensors that are usually used in like aquariums. So for regulating like the fishes environment. You can connect it to an Arduino and then get like live readout of the data and stick it in a mason jar, because this is New York City, I guess. And so yeah, that's also something that I've made here at the VFL and that you guys can see next door in action. And so the idea is to test on these different materials that we're making. Oh yeah, this was advertisement for this stuff. Oops. So I think this is really exciting that we can use this microbiological genetic information to inform the design of the built environment. So we've talked about how we can do that for bioremediation, but also I think it's really interesting, and architects and designers can tell y'all more about that. But to use design to communicate an understanding of this invisible but ubiquitous microbiological world that we interact with. And so we've drafted this design proposal of how we think we could visualize the microbiological activity that's going on in the canal. And so we've proposed these sensors, biosensors, that read things like pH, temperature. Those are like abiotic measurements. But if you measure dissolved oxygen and redox potential, that gives you an idea of the metabolic activity of the bacteria in the canal. And we've got a prototype working also, so we're excited to show you guys that. This is part of a much larger design proposal that we submitted to a design competition given by Access Civitas last year, which we won. Actually, we were super excited, and so I think one of the most interesting things about this project to me is how it's really multifaceted and multidimensional, and it's brought us together who have completely different backgrounds and different sets of expertise, but somehow a common goal. And I think that this kind of collaborative project, bringing together biology and design, is something that's happening more and more often. And so I've become part of this rising community of so-called biohackers, so people who aren't formally trained in biology, maybe they are formally trained in biology, but who are practicing biology in contexts that are not the the ivory tower academic context. And oftentimes they undertake projects that have particular interest to them because of the community that they're in or their particular interests. And so I just spoke at the very first Biohacker conference in Oakland last week. It was called Biohack the Planet. You guys can check it out and see what other biohackers are up to. But I think that these communities that are distributed but that have common interests and also specifically kind of the like fab community who tend to have be distributed worldwide but have access to the same resources gives this really awesome opportunity for collaborative and distributed citizen science. And so I think this project lends itself to that particularly well. And so we've come up with different ways that like different biohacker labs can work together. And so people can, because contamination is a ubiquitous problem, unfortunately probably everybody has their version of the Gowanus Canal in their backyard. And so everybody can do the same types of culture experiments, for example. That's super simple to do without access to any kind of controlled chemicals. We can share our process and design files. Maybe there's an optimal combination of material and design that works not only in the Gowanus Canal, but in many other places. And because it's easy to share design files, and because these digital fabrication tools are more and more readily available, then people can fabricate their own versions of these bioreactors. Also share sequencing data to see if we can find examples of convergent evolution. So bacteria that have evolved similar functions but independently, but because they were challenged with similar problems. I made a GitHub to share this. It's called Dystopic Solutions. There's nothing on it yet, but I'm planning for it. So it's open. So yeah, with that, I'd like to thank my lab at Cornell. It's my boss, Chris Mason, who never says no to anything. So that's why I was able to do an artist residency in the middle of my postdoc. And Ian and Matt, who are right here, and Ellen at GenSpace. And I'll take any questions.
My question was, to what extent are these bacterial communities like ever present and drawn by the certain conditions, and to what extent are they sort of regionally or locally defined?
It's a very good question. So we compared these sequencing data to a bunch of other environmental samples from around New York City, from the subways in particular, and also like environmental like soil samples, and they're significantly enriched. So the bioremediation related pathways are significantly enriched in the Gowanus Canal samples. So they've been specifically selected for in that environment.
Did they colonize the space because it was cool to live there, or did they actually evolve to live there? I mean, how did that work? Like, where it came from?
Yeah, that's a good question. They're probably bacteria that come from the soil or from other environments. So we saw that they were related to marine environments or to the human gut. And so the bacteria that are living there are probably related to the bacteria that were originally present in the soil. The ones that were able to survive and proliferate were the ones that were able to respond to that toxic challenge. And so these particular compounds, like complex hydrocarbons, atrazine, arsenic, things like that, they're not things that humans have invented. They're things that exist in nature, just distributed in a different way than the way that we have redistributed them. So this kind of contamination is like a redistribution of resources. We've concentrated in this environment elements that normally are much more distributed. So it's not unusual. It's not unlikely to think that are bacteria that already have these kinds of genetic pathways and that exist in nature, because these are compounds that exist in nature. It's just that the ones that were able to survive in that concentrated environment are the ones that have them.
There's this Dutch guy, Dutch microbiologist, I don't know, a couple hundred years ago, named Bos-Becking, who came up with the Bos-Becking hypothesis, which is basically that everything is everywhere, but the environment selects.
Mm-hmm.
So from the perspective of microbiology, these cells are, because they're single cellular organisms and they sporulate, they are essentially just distributed in the world, and their genes are distributed in the world. So really what determines their expression in the environment is their substrates. And that's somewhat of our thinking here is like, okay, here are these substrates. Now let's see if these organisms are on.
Mm-hmm. Yeah.
That's kind of related to what I was going to ask, is it's like, so the implication is that these things develop because there's some kind of toxin there that they like gobble up, or so what is the equilibrium that's like, if in a like the perfect environment or the equilibrium environment, what would happen? Like what would the environment be like? Is that been created in the lab?
If the toxicity would be able to disappear? Or?
Like, yeah, like if all these organisms reached equilibrium and there was no more interaction or whatever, what would be the mean state?
Mm-hmm.
Mars?
I think, so if each of these guys were to exist in isolation.
Yeah, like has that ever been isolated in a lab to determine, well, this is like what the world would be like if we did not interact with it?
Right. I think that's kind of a moving target, so it's hard to recreate. And the thing about environmental metagenomics or environmental microbiomes is that the populations are extremely diverse, and so their interactions are very complex, and also the environment that they live in is very complex. So it's pretty hard to tease out one by one each individual species and figure out what each one of its function is in isolation and what each one of its function is in combination with others. So there's been models that have been developed of that, in particular in human disease. So there's different human pathogens like staph, for example, that is known to have particular functions and respond to certain antibiotics, for example. But when there's a co-infection with two species, staph and another species, then all of a sudden it doesn't respond to the same antibiotics. It might be sensitive to others. And that's just the interaction between two species, and that's already super complex. But here we're talking about like 30 or 50 species. And so it's a tough question to be able to model that, picking out each member of the community all by itself. Which is why I think the approach of not doing like top-down engineering, but more like coaxing the population as a whole in a particular direction is more feasible. Because people do try to genetically engineer bacteria that like can eat oil, for example. And that's one particular function. And it's reasonably easy to get a bacteria to do one thing. It's harder to get a bacteria to do multiple things. And it's even harder to get a bacteria to do multiple things and then not be sensitive to the rest of the cocktail of toxicity.
Can you talk more about like making a bioreactor out of an Arduino? Because that's really interesting that like this thing that's $20,000+ you were able to kind of figure out like how to make that with Arduino. Did you have Arduino experience before that? So what was that process like for you?
Yeah, I mean, it's pretty revolutionary, I think. Actually, as a scientist, this idea that you can make your own instruments, like, I mean, up until now, we were completely bound to what the biotech companies could provide to us. And granted, their bioreactor is probably a lot more robust and stable than my Arduino bioreactor, but that I don't necessarily need something that's going to run for 3 months straight without, breaking down or anything. And I got into programming Arduinos maybe like a year ago or so, and I found it pretty intuitive. I have a background in computer science, so I'm comfortable with the programmatic interface, but I think that that's what made it really easy because electronics always seemed kind of intimidating to me. Like, I still don't know how to use a multimeter, but I know how to read data out through a program, and so yeah, it's definitely like opened my perspective as a researcher.
Is the EPA receptive to this sort of high remediation? Are they just like dredge and cap it?
Don't want to talk about this.
Definitely the latter. We tried opening a few sort of conversations that haven't gained much traction. So yeah, it's a matter of just dredging it, shipping it to New Jersey or somewhere, dumping it, I guess, in a landfill, and then sort of covering up the rest that's still looking at.
I mean, because in this context, the EPA is basically the Army Corps of engineers, and there's a lot of just ways that have worked, quote unquote, in the past that will continue. But I don't know. We'll keep doing our thing. They keep doing theirs. Hopefully, we'll converge.
This isn't happening on any scale in the United States at all.
What do you mean this?
These kind of projects? Yeah, I mean, the idea of using bacteria for bioremediation isn't a new thing, but the approaches that we've taken, and especially the sequencing and these particular experiments, as far as I know, are a new approach to it. I mean, I think the optimal thing is if we could get access to a little field station, because these kind of experiments are going to take years, if not decades. I mean, it takes a long time to assay these kinds of rates of metabolism that themselves aren't happening very quickly. So, I mean, if we could have like a little corner of the canal that we could use as a field station. Yeah, exactly. Then that would be awesome. And the thing is, this is a strong genetic heritage. Like, evolution is an optimization algorithm against. So the genetic content that we have today is the optimal solution after like 150 years of computation time. So I think it would be a shame to destroy it. And so I feel like the effort that we've been doing with just like cataloging this information, taking samples, maintaining them, is important unto itself as far as preserving the genetic heritage.
As an instrument, the bioreactor is more about measurement, or is it about creating or generating an environment?
Good question. So both, actually. So one function is kind of in the experimental context to be able to run a bunch of experiments in parallel and not alter the environment any more than what we're putting in as far as materials and stuff. Then we can also imagine altering it by tweaking the pH or other factors, temperature, just adding certain compounds that will oxygenate the water also, and then seeing if there's some of those alterations that.
So on one level, it's like isolating an environment that you've created, and on another level, it's like then censoring whatever happens in that environment.
Yeah. So having a controlled environment is super important as far as interpretation of your results, because you need to be able to reproduce those results many times. So if we were to be doing experiments like that in the canal, it'd be all over the place because there's the tide coming in and out, and rainfall, and sewage, who knows what. And And so one of the problems with environmental studies in general is that your subject of study isn't people.
Are the bacteria a bit like artists moving into the empty warehouses? Are they effectively gentrifying the sludge? So the question is, if you do the same study a year from now, will there be bacteria with little mustaches and funny hats? Will it be a different successional ecology? And what are the implications over the long term for the kind of bacteria we can expect in 5 years from now?
Yes, excellent question. As far as the colonization of hipster bacteria, I think that we can imagine that there will be a form of succession, because as each of these species is, or as each of set of species is in operation, they are altering their environment. So maybe once the composition or the concentration of toxin A has reduced enough, then other kinds of bacteria that are more susceptible to it are going to be able to come in. And actually that is kind of the idea behind creating these shapes. So these shapes are on like a millimeter scale, and bacteria exist on a micrometer scale. But I do think that it's important to create kind of these more macro scale shapes, because they'll effectively be creating these micro environments. And so you can imagine that in these smaller environments, you can have an accelerated process of succession. I haven't looked for mustaches though, but you can check it out in the microscope.
Are there any like leading candidates of shape and material that are?
So far, we've just set up the experiments, and so now we're waiting for results. Yeah.
That leads into my question. I was going to ask what what would you be excited to find out? What sort of quantified sort of results would make it feel like a success for you? And then on top of that, then what's sort of next in the process beyond sort of this residency?
Yeah. So if we could see any kind of differential rate of metabolism between different materials, for example, that would be fantastic. That would be a huge step in the right direction. And I'm particularly interested in concrete, actually, because concrete is one of the most widely used building materials, and but it's pretty toxic to the environment because of its high pH. And so if we could see any difference between the regular concrete and the bioreceptive concrete, that would be amazing, because then that could translate pretty directly into actionable information as far as design. And so then the next steps after that would be like scaling it up and making it big and doing really big like in situ experiments in the canal itself.
Is there a way to computer simulate?
Yes. Yeah, totally. Mm-hmm. There's various algorithms that have been developed to simulate like population dynamics and growth, and we haven't generated any of them actually in these shapes, but that would be a good idea. Mm-hmm. Yeah.
You could link that back to your 3D modeling kind of optimization.
Mm-hmm. Yeah, totally.
Why can they feed and thrive off of toxins that more complex organisms die?
Yeah, I mean, it's probably a question of necessity, and so if these complex hydrocarbons are the only or the most prevalent carbon source that's available, then all organisms need carbon to be able to synthesize new molecules, because we're like carbon-based organisms. And so it's been described in other contexts, like there's like weird like deep sea marine environments where you find bacteria that use really non-canonical carbon sources, and who have developed specific pathways for that. So the idea of adapting to their carbon source is something that has been seen in other contexts also.
There are hypotheses too. I think we should wrap it up.
Okay.
So, enough Q&A. There have been hypotheses too that these are some of the fundamental environments. That's where RNA came from. That's where DNA came from. With these deep sea vents of what we would perceive as like toxic oil volcanoes.
Yeah.
And that's actually potentially the original, the birthplace of all life as we understand it. So in that mode of thinking, the Gowanus Canal is just another environment. And that's kind of like what we tried to sort of approach this project with.
Yeah, there's all sorts of like natural extreme environments, like the deep sea vents, or extremely cold or hot, or there's these like methane craters that are constantly burning. Also,
Yeah, I mean, it's not like we're not actually making anything new as people. Right? We're not.
It's your analogy.
We're not really yeah, nothing's really that foreign. So, yeah, thanks.
The EPA has argued that bioremediation won't work in the canal because it's primarily superficial. It can only affect the top 1 or 2 feet of the sludge or the soil. And in your experiments, do you feel that there is actually bioremediation potential deeper down in the 20, 30 feet underneath ground? Is there life down there? Is there a change process going on? Is there actually a potential for bioremediation that's beyond just surface?
I wish we could get cores that would go that deep. That would be amazing. But part of the design that you guys can see next door is assembling these structures into much larger, like architectural shaped structures that would actually be able to sink deep into the sediment and, propelled basically by their own weight. And so in that way, we hope that we could access those deep layers and provide some kind of interface with those layers that are currently isolated.
Because I think I would be confident anyway that there's definitely some microbial action and potentially some remediative action happening as far deep as we get core.
Okay. Well, great. I would like to thank BKBioreactor and all of you for attending, and please head next door to see the work. And please grab another drink and some chips. And thanks for coming out during the rain.
Yeah.